Hello, my name is Jérémy Cours. I am PhD student at INRAE in France under the supervision of Christophe Bouget and Aurélien Salé. Today I decided to bring you in forest to talk about the short and mid-term effect of forest dieback at the habitats and saproxylic beetle communities. To begin, let's go by some context. Natural disturbances are highly important in terrestrial ecosystem functioning. They are major drivers of forest temporal dynamics and they shape the structural heterogeneity of forests. Multiple disturbance complexes generate key resources such as tree-related microhabitats, canopy gaps, and snags and logs, generally referred to as biological legacies, which are colonized by wide and often specific communities. Among them, saproxylic beetles are a highly diverse group of insects that depend on dead or decaying wood and play important ecologic roles by participating in carbon and nutrient cycles or by complexifying trophic chains. The ongoing climate change is directly and indirectly affecting natural disturbance regimes, such as droughts and heat waves by increasing temperature worldwide, but also pest outbreaks and storms. Warmer and drier conditions facilitate drought, wildfire and insect outbreaks. Lately, several large drought-related dieback events have been reported in forests on all continents. A further increase in drought-induced dieback and decline in terms of frequency, intensity and spatial extent is expected to arise. As a consequence, deadwood supply and the number of tree-related microhabitats are likely to increase, and this may favor saproxylic communities. To test this hypothesis, we use 3 study cases: one pest-induced dieback in spruce stands in Bavaria, Germany, and 2 drought-induced forest diebacks, first in fir stands in the Pyrenean mountains, and second in oak stands along the Loire Valley. We respectively implemented 19, 56 and 18 study plots in each study cases, along gradients of increasing dieback. Within these plots, we measure deadwood amount and its characteristics: diameter, stage of decay, position, lying or standing. We also identified tree-related microhabitats borne by living and dead trees, such as tree cavities, dead branches in canopy, or fruiting bodies of saproxylic fungi that are colonized by specific species. In parallel, we set up traps in the center of each study plot to sample flying active insects from May to September. Insects were identified in lab by experts using traditional morphological criteria. In the second part, I will present you our first results about the effect of dieback on habitats. We identified several important trends in dieback-induced habitat changes. First of all, deadwood significantly and highly increased in disturbed stands. In Bavaria, where experimental stands were classified as healthy or disturbed forests, we observed a 4.5 time increase in local deadwood amount. In fir and oak forests, we also observed a non-linear increase in the deadwood amount along the dieback gradient. Moreover, in disturbed stands, we could potentially observe changes in deadwood profiles, with higher volume of large and well-decayed pieces of deadwood than in healthy stands. For multivariate analysis, we observed differences in assemblages of deadwood along the forest dieback gradient, in conifer but not in the oak study case. As well, assemblages of tree-related microhabitats were significantly affected by dieback in all of our 3 study cases. Indicator value analysis showed that declining forests are overall characterized by higher occurrence of fruiting bodies of saproxylic fungi, bark loss, bark pockets, and bark shelter. As an example to understand the potentially ecological importance of these microhabitats, bark shelter could actually be used as a shelter by bats. In contrast, conifer healthy forests were characterized by higher occurrence of epiphytic bryophytes, which are known to be more dryad communities, thus mainly associated to closed and mature forests. In the third part of this video, I will present you our first results about the effects of forest dieback on saproxylic beetle communities. The increase in deadwood amount, as well as shifts in tree-related microhabitat profiles in disturbed forests, did induce changes in saproxylic beetle communities. In oak stand from the Loire Valley, we measured a positive response to the forest dieback of the overall species richness of saproxylic beetles. We also observed contrasting response of guilds to forest dieback according to their trophic specialization. Nonetheless, further analysis on a wide range of crown-dwelling insect guilds are in progress. In spruce Bavarian forest, the pest-induced dieback led to sharply disturbed stands. Although we did not observe significant responses of the total abundance and species richness, we noticed significant positive response of red-listed species in our models. Nevertheless, total species richness positively responded to standing deadwood, which increased in disturbed stands. Moreover, we also found higher abundance and species richness of flower visiting or cavicolous beetles in disturbed forests. Finally, we also demonstrated that communities in disturbed forests have preference for larger and more highly decayed deadwood through an increase in community-weighted means of related ecological traits. In silver fir Pyrenean forests, progressive drought-induced forest diebacks led to small discrete disturbed patches. In this study case, we carried out an in-depth analysis of the effects of both local and landscape scale. From our results, taxonomic and functional diversity, but not phylogenetic diversity, significantly benefited from forest dieback at both local and landscape scale. Moreover, we noticed a specialization in more extreme values of the functional space in response to the increasing forest dieback at the landscape scale. This will be a positive result within the worldwide context of biotic homogenization in man-disturbed ecosystem. Forest dieback also had positive effect on xylophagus species as well as fungicolous and cavicolous guilds. It indirectly suggests higher densities of fruiting bodies of saproxylic fungi and rot hole cavities in declining forests. Besides, it seems that healthy forests have benefited from the colonization of fungus and cavity-dwelling species from disturbed forests through spillover effects. Our results support a habitat amount hypothesis type of saproxylic beetle response to forest dieback. So it's time to conclude. Our analysis revealed a positive response of saproxylic beetle diversity to forest dieback in 3 European study cases. At this point, it should be underlined that conventionally managed forests are known to host significantly less deadwood and tree-related microhabitats and degraded deadwood-associated communities compared with natural forests. As a consequence, we could consider unharvested declining forest stands as relevant sites for biological conservation, at least temporarily, because they favor the functional diversity, abundance, and richness of saproxylic beetle species. However, since climate change is ongoing and forest dieback are increasing, we need further data about longer-term effects. I thank you very much for your attention.